Good day everyone, today we'll be discussing gene therapy. So, as a review, genes are carried out on a chromosome. It is the basic units of heredity, but when it is altered, it can cause dysfunction or improper function of a protein. So when there's a mutation in a gene, it will change the codon, which will change the amino acids, and then changing the conformation of the protein and its function. So genetic disorders result from mutations in the genome. So what is gene therapy? So it is a technique for correcting defective genes. So there are four approaches. So a normal gene is inserted to compensate for a non-functional gene. So this is the most common approach. An abnormal gene is traded for a normal gene. So the abnormal gene would be swapped by homologous recombination. An abnormal gene is repaired through selective reverse mutation, and it would cause a return to normal function and then change the regulation of the gene pairs. So it controls the expressions of genes similar to epistasis when one gene affects the expression of another gene. So in the 1980s, scientists began to look in gene therapy. They would insert human genes into a bacterial cell. Then the bacterial cell would transcribe and translate the information into a protein. Then they would introduce the protein into the human cells. So the bacterial cells would be the vector or the carrier of the human gene. So the first gene therapy was performed on September 14, 1990. So Ashandi de Silva was treated for SCID or the severe combined immunodeficiency. So the doctors to remove her WBCs, inserted the missing gene into the WBC and then put them back into her bloodstream. So with that procedure, they strengthened her immune system but only worked for a few months. So how it works? So a vector delivers the therapeutic gene into a patient's target cell. The target cells become infected with the viral vector. Then the vector's genetic material is inserted into the target cell. Then the functional proteins are created from the therapeutic gene, causing the cell to return to a normal state. So a vector is a carrier molecule, usually virus. The target cells are usually the liver, the target cells are usually in the liver or the lung. So gene therapy is done by using a vector to insert tiny fragments of DNA into a disease cell's nucleus. A vector is simply a shuttle that used to carry DNA into a cell and then leave it there for a cell to use. Generally, the vectors used in gene therapy are viruses. A virus can hold onto the DNA as it enters the cell and then deliver the DNA to the cell. The vector is then injected into a person's body directly, or it can be mixed up with some of the person's cells outside the body that are then replaced. Once the vector is delivered to the target cell, scientists hope that the cells will absorb the new functional gene and integrate it into the cell's own genetic makeup. Once this occurs, the inserted gene should function normally, creating the necessary protein that were deficient before the therapy. The patient should become healthy as their new genes begin to work normally. So a healthy gene was inserted in a virus. For example, this is a retrovirus. Then we have here the unhealthy cells. So the retrovirus will deliver the healthy genes to the affected cells. So viruses, they replicate by inserting their DNA into the host cell. So gene therapy can use this to insert genes that encode for a desired protein to create the desired trait. So there are four different types of viruses used for gene therapy. We have retrovirus, adenovirus, adeno-associated virus, and the herpes simplex virus. So retrovirus, they are double-stranded DNA copies from RNA genome. So retrovirus goes through reverse transcription using reverse transcriptase and RNA. So katulad siya ng mga SARS-CoV, coronavirus 2. So the double-stranded viral genome integrates into the human genome using integrase. So integrase insert the gene anywhere because it has no specific site. So it may cause insertional mutagenesis, so one gene disrupts another gene's code. So vectors are derived from the human immunodeficiency virus and are being evaluated for safety. Adenoviruses, they are double-stranded DNA that causes respiratory intestinal eye infections in humans. So inserted DNA is not incorporated into the genome, not replicated, has to be reinserted when cells divide. So example ng adenoviruses ay common cold, na nagkakos ng common cold. Next, we have the adeno-associated viruses. So they are small, they single-stranded DNA that inserts genetic material at a specific point on chromosome 19. So unlike other viruses, it has a specific point where they will insert the genetic material. So they are from the parvovirus family. So the parvovirus causes no known disease and does not trigger patient's immune response. So low information capacity, gene is always on, so the protein is always being expressed, possibly even in instances when it isn't needed. Example ay hemophilia treatment, so a gene carrying vector could be injected into the muscle, prompting the muscle cells to produce factor 9 and thus prevent bleeding. So a study made by Wilson and Carey High at the University of Pennsylvania. So once inserted with the gene carrying vector, the patients have not needed factor 9 injections for more than a year. We also have the herpes simplex virus. It is a double-stranded DNA that infect neurons. So example is the herpes simplex virus type 1. We also have non-viral options. So direct introduction of therapeutic DNA. How? Only with certain tissues and requires a lot of DNA. So creation of artificial lipid spear with a Quayle score like liposome. So liposome, it is a spherical vesicle having one lipid bilayer, and it can be used as a vehicle for administration of nutrients and pharmaceutical drugs. So we also have receptors. So DNA is engulfed by a cell membrane, but it is less effective. We also have trying to introduce the 47 chromosome, exists alongside with the 46 chromosomes that are already present in our body, and it could carry a lot of information. So the current status, the FDA has not approved any human gene therapy product for sale. Reasons, because in 1999, Jesse Gelsinger died from a multiple organ failure 4 days after the treatment of OTC or the ornithine transcarboxylase deficiency. So that was triggered by severe immune response to adenovirus carrier. So in January 2003 naman, how to using retrovirus vectors in blood stem cells because children develop leukemia-like conditions after successful treatment for X-linked severe combined immunodeficiency disease. Problems with gene therapy, they are short-lived. So multiple rounds of therapy is needed, the immune response. So new things introduced in our body leads to immune response, so increased response when a repeat offender enters. Viral vectors, so patients could have toxic or immune or others inflammatory response to the vectors that are inserted in our body. We also have multi-gene disorders like heart disease, hypertension, Alzheimer's, arthritis, and diabetes are hard to treat because you need to introduce more than one gene. So it may also induce a tumor if integrated in a tumor suppressor gene because of insertional mutagenesis. So unsuccessful gene therapies, as I said a while ago, Jesse Gelsinger died in 1999 due to the gene therapy, who lacked the ornithine transcarboxylase. So OTC deficiency is a genetic disease that prevents body from breaking down ammonia, which is a metabolic waste product. So the excessive buildup of ammonia often cause death soon after birth unless the patient's diet is immediately adjusted and monitored throughout the entire life. So Jesse lived on a strict non-protein diet and he controlled his OTC fairly well. So Jesse volunteered for a gene therapy experiment designed to test possible treatments for OTC. He thought he could help newborns afflicted with OTC by being a volunteer in the experiment. He enrolled as a subject in a gene therapy experiment in which vector carrying a normal OTC gene was injected into his liver. The vector being used to deliver was adenovirus, which is a modified version of the virus that causes you to get cold. For most part, Jesse was informed subjects had received adenovirus without serious complications, but for some reason, Jesse was different. After injection of the vector, a fatal reaction was triggered, and then 4 days after the injection, Jesse died. So after the doctor shot the normal OTC gene to Jesse, he developed a high fever, his immune system began ragging out of control, his blood began clotting, ammonia levels climbed, his liver hemorrhaged, and a flood of white blood cells shut down his lungs. So one problem with gene therapy is that one does not have control where the gene will be inserted. So it is called the position effect. So the gene regulatory aspects are always uncertain after gene therapy. So we also have the unsuccessful therapy for severe combined immunodeficiency. So they have profound deficiency of lymphocytes. So it is an inherited X-linked recessive disease that affects boys. So autosomal recessive siya, and there are several abnormalities in the immune system when the defective gene is encoded on an autosome. So SCID are due to the deficiency of adenosine deaminase. So peripheral T cells were transduced with a vector bearing the gene for adenosine deaminase. So the experiment was extremely labor extensive because mature peripheral blood T cells were modified rather than stem cells. We also have successful 1-year gene therapy trial for Parkinson's disease. Neurologics, a biotech company, announced that they have successfully completed its landmark phase 1 trial of gene therapy for Parkinson's disease. So this was a 12-patient study with 4 patients in each of 3 dose escalating cohorts. So all procedures were performed under local anesthesia, and all 12 patients were discharged from the hospital within 48 hours of the procedure. So primary outcomes of the study design, safety and tolerability were successfully met. There were no adverse events reported relating to the treatment. So the gene transfer procedure utilized AVV, or the adeno-associated virus vector. So it has been used safely in a variety of clinical gene therapy trials. So in its Parkinson's disease trial, Neurologics used its gene transfer technology. So recent development, genes get into brain using liposomes coated in the polymer called polyethylene glycol, a potential treatment for Parkinson's disease. RNA interference or gene silencing are also used to treat Huntington's. So siRNA is used to degrade RNA of particular sequence. So siRNA, or the short interfering RNA, or the silencing RNA. Creations of tiny liposomes that carry therapeutic DNA through pores of nuclear membrane. Sickle cell disease was also successfully treated in these. So future prospects in gene therapy for blood, treatment of sickle cell disease, hemophilia, and severe combined immunodeficiency. For metabolic diseases, we have glycogen storage diseases, lysosome storage diseases, and phenylketonuria. For muscular, we have Duchenne muscular dystrophy, myotonic muscular dystrophy. For lung, we have cystic fibrosis, cancer, brain tumors, breast, colorectal, malignant melanoma, ovarian cancer. Cardiovascular, we have atherosclerosis and hypertension. Infectious diseases like AIDS and possibly other viral diseases that involve latent infections.